Well, hi, thank you for coming out to hear me, and thank you for my own little fan club over there. When Luan invited me to give this talk last year, maybe October, something like that, he sort of whispered in my ear, and by the way, you know, maybe you could talk about the hype versus hope, because after a lot of these talks, audience members come up to me and say, well, when am I going to get that breakthrough? I don't see any of these breakthroughs that are anywhere near me soon. So I gulped and thought, that's a big order, because I'm a little bit culpable myself, having written 2 books about 2 medical revolutions, which have yet to really sort of appear at a doctor's surgery near you. And what can I say? You know, as a journalist, we have to tell stories, and, you know, inevitably a little bit of hype gets in there, and when you are telling a story, as much as one tries not to. I had sort of prepared myself to give a sort of half-empty, half-full type of story, saying, well, even though stem cell therapy hasn't made a big splash yet, and even though you're not going to your specialist with your genome on a computer stick, just give it time. It's sort of working its way through, and it's helped the toolbox of medical discovery, and things are coming. So that was the talk I was going to give you, until something happened. A bit of serendipity got in the way. I was asked to be on a panel at a conference called Above All Human, and it is a conference for ethical startups. It was held at the exhibition buildings, I think it was last October, September, something like that. And all you know, hundreds of ethical startups congregate, and they talk about what they're doing. And I was asked to be on a panel for Eugene, which is a genetic testing company started by Zoe Milgram, co-founded by Zoe Milgram and another fellow. And Zoe Milgram is a former genetic counsellor. So she asked me to be on the panel with her as the science journalist. And I also met this woman, Megan Donnell. Is a very vibrant, appealing person. She looks like a really happy person. And when I heard her story on the panel, I was completely devastated to hear her story. So Megan introduced, she's not a scientist, but she pulled back the curtains for me on the gene therapy revolution that had arrived and I hadn't noticed. She was a member of the public who was living the impact of gene therapy. And how had I missed this? I don't know. I was at the time the editor of a science magazine. All my editors, all my writers missed it. Well, I'll come back to that later. And as you can see, Megan is a vibrant, appealing person. What you can't see is what is simultaneously her life's greatest tragedy and her life's greatest mission. So this is Megan with her husband, Alan. Sadly, they parted about 8 months ago. And her 2 gorgeous children, Isla, who's now 10, and Jude, who's 8. And they were born healthy, beautiful children. But when Isla was 2 and the second one, Jude, she took him to the nurse's health centre for a checkup. The nurse mentioned, you know, are you a little bit worried about Isla? Because Isla wasn't really putting words together into sentences, as 2-year-olds are starting to do. And this started Megan on a diagnostic odyssey, which it took 2 years to really uncover what was going on with Isla, as she, from then on, from the age 2, continued not to meet her developmental cognitive milestones. And finally, she got a diagnosis when she was 4 years old. She has Sanfilippo Syndrome, which is a very tragic diagnosis. Even more tragic, her 2-year-old also has been afflicted with it. She had no ideas that she and her husband were both carriers. And what this means is that the children will progressively become more brain damaged as they grow older. By the time they're in their very early teens, they'll become paralysed, unable to walk or talk, and their expected lifespan is somewhere in the mid-teens. So Megan got this devastating diagnosis and was basically told, don't hold any false hope. This is what you're in for. Let me explain a little bit more about what this disease is. It's a maddeningly simple disease. So what's going on? The children have very high levels of a sugar called heparan sulfate. And I liken heparan sulfate to the weave on a tapestry. It lies there between cells and creates a matrix between cells. And what it's doing is holding in place proteins on that matrix in the same way that this tapestry is holding motifs. So here we are, we've got the heparan sulfate weave holding these proteins in place here. And this tapestry needs to be constantly unpicked and new proteins put in there. What that means is you have to be able to excrete the weave, the heparan sulfate chains. And these children lack the enzyme to break down the heparan sulfate sugar, missing just one gene for one enzyme. If only you could replace it. And indeed, the early treatments have involved just injecting these children with the enzyme, but the tragedy is it can't get past the blood-brain barrier. It can't get to the brain where the brain is becoming slowly poisoned by these heparan sulfate sugars. However, Megan Donnell did not listen to her doctors who told her not to hold any false hope. She and a friend got onto the internet and started doing some research, and they discovered gene therapy. The wonderful thing about this gene therapy is that it looked like it could send the gene across the blood-brain barrier and start to impact the toxic levels of this sugar that were building up in the brain. So just to give you a little crash course on gene therapy, which I'm guessing you're not all intimately familiar with, the whole idea really is very simple. You're missing a gene, you replace the gene. How do you replace a gene? Certain viruses have evolved to carry DNA into our cells, so we make use of them. Here is the virus here. It's in a nice little capsule, like a space capsule. It's icosahedral, a beautiful thing to look at. It carries its own little strand of DNA, but you can put in the gene that you would like it to carry into the body. So you stick that gene into the virus, and then you put it in the syringe and you inject it. It goes into the body, and some of it does appear to get into the brain. Once it's in the brain, this capsule releases its DNA into the brain cells, and the DNA just sits there and starts producing the missing enzyme. So I'm not able to tell you whether either of Megan's children have been in this trial, but I can tell you that as of November 2018, 14 children have been treated with increasing doses. They received a single intravenous injection of this, what's called the AAV9 vector containing the faulty gene. And in all of the children, they've seen when they look in their blood, their cerebrospinal fluid or their urine, that the sugar levels are coming down. They're starting to see encouraging neurocognitive signals, meaning the children are not deteriorating at the rate you would expect. And the really encouraging thing is that when you get the very young children, they are the ones who've had the least damage, and you're starting to see effects. So according to anecdotal evidence, in a child that was just a little over 2 years, unlike Isla when she was 2 years, they were putting adjectives and adverbs into complete sentences. Could it be a cure? It's not a vain hope. And this is why Megan is so excited about gene therapy, because miracles are happening. Somehow I missed it, but this is a case that was reported in 2017 in the New England Journal of Medicine. 15 children who were born with the disease called spinal muscular atrophy were given gene therapy. Now, in the normal course of events, for those of you who aren't familiar with this terrible illness, these children in the first few months of life, instead of their motor neurons, which connect their muscles to their brain, instead of those motor neurons sprouting and connecting up the muscles to the brain, they wither and die. So the children become increasingly floppy, and By the time they're about a year old, they're starting to get paralysed, unable to eat or move around, and they generally die somewhere between 15 and 2 years of age. But 15 children were given an injection containing according to the same protocol I showed you before, with this adenovirus containing the missing gene. And here we have an example of one of the children, Evelyn Villarreal, at 3. As you can see, she shouldn't be alive. Not only is she alive, she's not paralysed. She's walking, she's talking, she's moving around. Another miracle. Children born with a form of retinal dystrophy, or it's a progressive blindness that starts to kick in the same way. A few months after they're born, the cells of their retina start degenerating, and they progressively become blind. They end up with tunnel vision, and they have no night vision. Again, gene therapy. This young woman was treated with gene therapy and her sight was restored. In this case, the product is called Luxturna. It was approved by the American FDA in December 2017. It was in fact the first gene therapy, in vivo gene therapy, I'll explain what I mean later, where it's injected directly into the person, that was approved by the FDA. And this company that produced it was a little startup called Spark Therapeutics, and just this February, Roche bought it for $4.8 billion. A third miracle. Some of you may be familiar with children born with severe combined immunodeficiency called SCID. They are confined in a bubble for life because they have no immune system. A common cold could kill them. And again, gene therapy, this time delivered ex vivo, where the child's bone marrow cells were taken from the child and the virus delivered to the bone marrow cells in the test tube, then re-injected back into the child and reconstituted a whole immune system for the child. That's 3 miracle cures. And it has prompted certain people to talk about biblical cures. You know, the blind are seeing, the lame are walking, children are getting out of their bubbles. Just as Gustave Flaubert predicted in 1856. But he didn't think it was going to be a miracle. He predicted that science would accomplish this. And he was right. How did I miss the gene therapy revolution? So I was busy at Cosmos magazine producing all these wonderful issues with the top stories on the cover. This issue, which we did in 2016, probably has something to do with it. Build your own baby. We were all dazzled by the CRISPR revolution. So it's a good moment to talk about the hype cycle, because I'm still wondering how I missed the gene therapy revolution. And I don't know if you were all aware of it and up with it, but most people I talk to certainly aren't. When I talk about the gene therapy revolution, they all just say CRISPR. But what seems to have happened, I don't know if you're familiar with this hype cycle. It's paraphrased by Roy Amara. We tend to overestimate the effect of a technology in the short run and underestimate the effect in the long run. So it seems that gene therapy had gone through its peak of inflated expectations, which was kind of in the 90s. It was down here in the trough of disillusionment with rather poor visibility. On the other hand, CRISPR was right at the peak and peak visibility. And with good reason. CRISPR is a revolutionary technology. The idea is not that you package a whole gene into a virus and send it in to replace the missing function. You use this editing tool like you might use a find and replace function on Word. What this can do is it can find the part of the DNA chain that is faulty and replace it with the correct version. It's easy, it's cheap, and it's precise in a way that gene therapy isn't, but we'll get to that later. But it's not as precise as we thought. There are what are called off-target effects. So it turns out you may be targeting this region, but the genome is a very long chain of DNA, 3 billion letters long. It'll stretch a few times around the Earth, I think. So you do get off-target effects that we can't always identify. Whatever its miraculous properties, CRISPR is only just now entering the clinic. Except for He Jiankui, who did a roguish thing and used CRISPR to make twin baby girls, to make them resistant to HIV infection. The whole world has really come together to condemn what he did because of these off-target effects. We don't know what the consequences are going to be for these two little girls who, by all accounts, it seems he wasn't bluffing that these two girls have been born. It's a very serious thing that he's done. It's called for a global moratorium, which was in place before, but I guess we're always going to have rogue scientists. Nevertheless, let's get back to gene therapy. What I'm showing you here looks like an explosion, and it is. There is something like 750 trials of gene therapy going on at the moment, and we already have 4 of them on the market, and more of them coming close behind, and all these things out in the pipeline. What we have already approved and on the market, it's gone through this incredible gauntlet where you have to do the clinical trials and make your way through the regulatory thicket. It is no small thing to get a drug to market. It is no small thing to get a gene therapy drug to market. This is quite remarkable. But I didn't notice. Since 2012, that was the first one that was approved by the European Union, the European Medicines Agency. It was a gene therapy called Glybera to treat lipoprotein lipase deficiency, an extremely rare condition where you can't break down triglycerides and you end up with creamy blood full of triglycerides. The next one was 2016, Strimvelis from GlaxoSmithKline, a big pharma, and this was to treat these children with severe combined immunodeficiency. Interestingly, this was also the very first gene therapy to go into a clinical trial in 1990. So 26 years later, from the first clinical trial to get to market. Hopefully the CRISPR technologies won't take 26 years. The very first gene therapy to be approved by the FDA was in August 2017 from Novartis. It's CAR T cells for acute lymphoblastic leukemia. I'll come back to that soon and explain what that's all about. And then a few months later, Luxturna, which was the therapy for the congenital blindness that I showed you before. So gene therapy is here, it's made it. 4 of them are already on the market. You know, there's tons of them in the pipeline coming. It's a good time to look back and see how did it get here, what have been the ups and downs along the road, especially since I missed it. Well, actually, as I said before, gene therapy is a kind of obvious idea and a simple idea. And in fact, it's been obvious since the 1960s when Marshall Nirenberg first cracked the genetic code. So yes, we got the DNA double helix in the 50s, but even though we got the structure of DNA and we knew it was some sort of a code, we didn't know how to crack that code. We knew it was a string of 4 letters, AGCTs, but what the hell did that code mean and how did you crack it? We kind of knew it was a code for proteins, but how did this code work? So Marshall Nirenberg in the 1960s, 61, cracked the code, and it was obvious to him where this was going to go. This knowledge will greatly influence man's future, for man then will have the power to shape his own biological destiny. And as the New York Times put it, the science of biology has reached a new frontier, leading to a revolution far greater in its potential significance than the atomic or hydrogen bomb. A simple idea, the path is clear, but many tragic missteps. The first tragedy was 1999. So this young man, Jesse Gelsinger, suffered from a genetic disease called ornithine transcarbamylase deficiency. He lacked a gene for a single enzyme that would break down urea. But the bottom line was he developed high levels of ammonia in his bloodstream that, when he was just a baby, sent him into a coma. When they figured out what was going on, they realised he had this genetic disease. But he suffered from a mild form. He got to the age of 18 by a combination of medication and watching his diet, not too much protein. But he was a fantastic young man by all accounts, and he volunteered for this trial of gene therapy to have this gene replaced into his bloodstream. So at the time, the virus they used was an adenovirus, the same virus that causes the common cold. They popped the missing gene into it and using the same technique that I showed you before, injected it into him. 4 days later, he was dead. When you do gene therapy, you have to inject huge amounts of the virus, far more than you would inhale with a common cold, straight into your bloodstream. And his immune system went crazy. He suffered from an inflammatory storm. So you get sick when you've got a cold. Imagine that amplified thousands and thousands of fold. All his organ systems failed, and he died. It was an incredible tragedy, and it set the gene therapy field back enormously. People basically stopped what they were doing, stopped their projects, and went back to the drawing board. And then another tragedy came. In this case, it was treating children who have severe combined immunodeficiency. It's a slightly different variation than the one I showed you before. It's called X-SCID, and it only affects boys because it's on the X chromosome. This is an example of a little boy, David Vetter. NASA made him a spacesuit so he could go home and visit. And he actually lived to be 13 until he had a bone marrow transplant from his sister and got out of his bubble and died just a few weeks later. Unfortunately, his sister had a virus that infected him. However, in this trial, not David Vetter, but other boys like him, they took a retrovirus and they took cells from the boy's blood, bone marrow stem cells, infected them with this virus, a different kind of virus called a retrovirus, which will actually insert its DNA into the DNA of the stem cell. And that's important because stem cells multiply. And so you need the virus to have put its DNA into the very DNA of that stem cell, or it'll get marooned. So it was successful. 20 treated children were all able to get out of the bubble, I believe, because they had their immune systems reconstituted. But the flip side of a virus that inserts its DNA into your DNA is that it can land anywhere in your DNA. And tragically, it landed near some cancer-causing genes and activated them. And 5 of these 20 children developed leukemia 5 years after the therapy. Most of them actually were treated and did not die of that leukemia, whereas they would have died probably of their severe combined immunodeficiency. So it set the field back enormously. The scientists went back to the drawing board, and what did they do? They did some serious engineering. They re-engineered these viruses. And I heard recently on a quiz show on TV, I think Hard Talk, the question was, how long was it in the time between the first powered flight of the Wright brothers and when we put 2 men on the moon with Apollo 11? And it was 66 years. That's not bad going then. When you think it's taken 20 years to re-engineer these viruses to make them safe. So adenovirus, the one that caused an immune storm and killed Jesse Gelsinger, we don't use that anymore because it's just sends our immune system haywire. Instead, we found it has a nice associate over here called Adeno-Associated Virus. And this guy seems not to bother the immune system too much at all. So this is actually the virus that was used to produce these amazing results in the children with spinal muscular atrophy. And I believe it was this sort of virus that produced the results with the blindness as well. So this is the favourite sort of virus now we're using because it doesn't rile up the immune system. And the genetic engineers have become incredibly clever. What they can do, I guess you can see this reasonably well. Well, you know, it lives in a capsid like this, a capsule. And what they can do is they can do some cross-dressing and change the capsule that it wears. And so they can tweak both which tissue it's going to target to, because for some diseases you want it to go to the brain, for some you want it to go to the liver, so on and so forth. So that all relies on the coat it wears. So you can do cross-dressing. The other thing they've been doing is working with these viruses that integrate into your DNA, the sort that you have to use if you're working with bone marrow stem cells that multiply up so much and will create a new immune system, for instance. So now instead of the retrovirus that inserted and turned on cancer genes, now they're using lentiviruses, and they appear to be much safer. They don't jump in next to cancer-causing genes. And they've also been able to tweak the genetic controls on these so that they are less powerful and less likely to inadvertently turn on surrounding genes. So not bad for 20 years. And just a bit of the highs and lows of gene therapy for these bubble boys, ex-SCID. So this was the low when 5 of the boys developed leukemia. But since then, we've been climbing steadily upwards with these re-engineered viruses. So that was the bad one. But in 2014, with a re-engineered virus, they were reporting 7 out of 9 boys got out of the bubble cancer-free so far. April 2019, just a week or so ago, using a different kind of virus, lentivirus, 7 kids out of the bubble so far cancer-free. The scientists have become very chastened. They're not going to say there's no risk, but when you're dealing with a disease like this, where the children are confined to a bubble for their whole life and very likely to die if the bubble gets pricked or something like that, I think it's justified to take this sort of a risk. And so far, it looks like they've figured out how to tame these viruses. So what are the next cabs off the rank? The first cabs of the rank, interestingly, were extremely rare diseases. So Glybera, this one for the disease where you end up with creamy blood because you can't break down the triglycerides, 1 in a million people. Strimvelis for SCID-ADA, 1 in a million births. Luxturna, the one for congenital retinal blindness, there are maybe 2,000 cases in the US. What are these companies thinking? You know, this doesn't sound like, would you invest in a company that was making cures for 1 in a million? This is not the sort of traditional business case that drug companies are convinced by. They've done their research and they can see what's coming. And the reason they have picked these incredibly rare diseases, there's a reason. Because they're so rare, because there's no treatment out there, because nobody would normally try and treat a disease like this or develop drugs for it, what they've been able to do is really fast track these treatments through the very ponderous and snarly regulatory systems. The FDA, the European Medicines Agency, have sort of come and met these companies halfway. Yes, come on, we'll help you get these through, to get these to the clinic. And the other thing is, when you're dealing with such rare diseases, you only need to do tiny clinical trials. Some of these clinical trials, if you've noticed as I've been going through, are only 15 people, 15 children, 6 children. So they're not as expensive as the sort of clinical trials we're used to for cancer that require, thousands of people before drug regulators are going to give them a look at. So that is the kind of logic of what's going on. They've gotten these guys through. Now it is time for the big game. They're scaling up. And we've already got the first big game type of gene therapy. I'm sure some of you will have heard of CAR T cells. It's been in the news a lot lately because Greg Hunt has just said that the government will pay for these treatments and they will be given at the Peter Mac in Melbourne. So let me tell you what CAR T cells are, in case you don't know what they are. So this ferocious looking brown thing here is a killer T cell, and it's attacking this cancer cell. So these are natural killers. They really ought to be killing our cancers better than they do, but sometimes they just don't notice the cancer cells. The cancers have a way of hiding themselves and foiling these T cells. So what CAR T cells do is they get equipped through gene therapy with a homing device to go and find these guys. CAR stands for chimeric antigen receptor. These were approved by the FDA in August 2017. So Novartis, big pharma now we're talking about, sells CAR T cells as a treatment. It's called Kymriah. It has been approved by the FDA for treatment-resistant acute lymphoblastic leukemia, ALL. In December 2018, our TGA also approved it, and just a couple of weeks ago, our federal government says it will pay for these treatments, which is good because they cost $500,000 a treatment. You think that's a lot? Wait. How does this work? So if you need it, you will be going to the Peter Mac. They will collect your T cells by a process called leukapheresis. And then they will, for the time being, send them off to the east or west coast of America to a Novartis centre where they will infect them with a retrovirus, a lentivirus actually, that carries a gene which will act like a homing device. Then they will send those cells back to the Peter Mac. It will altogether take about 5 weeks. The Peter Mac will expand the cells in its lab and they will re-inject them into the patient. And yes, the government will pay. And what's coming soon? Haemophilia B and A, this is the absence of clotting factors 9 and 8. Now we're starting to talk big markets. 400,000 people globally. It's a crowded space. We have lots of drug companies now developing gene therapy here for haemophilia B and A. Pfizer collaborating with Spark. So we've got big pharma in there. Beta thalassemia, where you're not making enough hemoglobin and you're constantly in need of blood transfusions to keep you going. Again, about 288,000 globally, 2 companies pursuing that, and they should be coming soon. Okay, I have never seen prices like this for medicines. One of my favourite journals, the MIT Review, had this on the cover just a few months ago. $2 million would save her life. Could you pay? Should you? So they might have been referring to spinal muscular atrophy, one of the miracle cures I showed you early on, where these children would be dead by the age of 2 but now are living. So Novartis, a big pharma, has recently bought the little company that makes this drug, and they say they're going to charge $4 to $5 million for a treatment. Luxturna, the treatment for retinal blindness, $425,000 per eye. So if you want both eyes, $850,000. The treatment for beta thalassemia, probably $1 to $2 million. How is this going to work? Maybe it's not so crazy. So this is what the drug companies are arguing. So it's kind of like the difference between monthly rent. So if you've got, let's say you've got haemophilia. So anyway, let me just finish what I was saying. The difference between monthly rents for a lifetime versus paying off your mortgage, which should eventually finish, because this will keep going. A scientist gave me this as an example. If you look at treating somebody with haemophilia, at the moment, you treat someone with haemophilia by giving them injections of the clotting factors, and it costs about $250,000 per year. And so over their lifetime, let's say they live 70 years, that's a cost of $17.5 million. That doesn't count the hospital costs, because somebody with haemophilia periodically gets hospitalised for bleeding in the joints and so on and so forth. So it's starting to look not so crazy if you are going to charge $1 million for what will be a one-off treatment. And if I haven't said that before, that is what we're talking about with gene therapy, because like Marshall Nirenberg realised in 1961, you're giving back a person the piece of the code that doesn't work, and they should be fixed forever after that. So what we're talking about is one-off treatments. It starts to sound like not such a preposterous idea. And what the companies are talking about, because they don't know yet, because this is such early days, the children, for instance, who've received gene therapy for spinal muscular atrophy, I think they're probably about 5 years old now. They have a lifetime to go. We don't know if this gene therapy really is going to be a one-off treatment that will cure them for life. Nobody knows the answer to this yet. So what the drug companies are saying is that, look, we'll do a sharing risk model where you give us an upfront payment. Let's say if we're talking about beta thalassemia, $1 million treatment, give us 20% upfront for the first treatment, and then only pay us 20% each year as long as it works. Now these are the kind of arguments that are going through. Okay, where's Australia in this game? We're in it. We're in the game. In Perth, Elizabeth Rakoczy at the University of Western Australia has been doing a trial of 24 patients with macular degeneration, treating them with gene therapy to try to stop the aberrant formation of tiny blood vessels. It's an anti-VEGF DNA, but don't worry about that. In Melbourne, as I've told you, the Pediatric is very involved with trials of CAR T cells, the killer T cells that can, through gene therapy, are given a guided missile to go and attack your cancer cells. John Raskow in Sydney is a clinician researcher. He's very actively involved in overseas trials, doing some of those trials in Australia. So we've been part of the beta thalassemia trials. That trial that was reported just last April resulted in cures for 15 out of 22 patients. In Adelaide, David Parsons is working on cystic fibrosis. I can talk about that later. We haven't had much success yet with cystic fibrosis, but I can discuss that later if somebody wants me to. But the real big guy in Australia is Ian Alexander at Westmead. I'd never heard of him until I started doing this story. He's a paediatrician, a very dedicated paediatrician, and he's also a toolmaker. And he's really in the game. He's been designing these vectors, which is where all the action is, which is where all the intellectual property is, going from the biplane to the rocket that gets the moon, redesigning these vectors so that they go to whichever organ you want to and do what you want to. So he's right in the thick of that. And he's trying to design vectors for pediatric liver disease. He's very involved in an SMA trial at the moment going on in Sydney. And he's also involved in a trial in the UK using vectors that someone in his lab designed for ornithine transcarbamylase, the disease that Jesse Gelsinger had. So Australia's in the game, and Ian Alexander is really in the race. He's chafing at the bit, and he would very much like the government to invest in this so that he can really start building the facilities to get Australia into the race. And what this requires is probably $10 to $100 million. I notice the chief scientist is here, but that's all right. There's a Chinese wall between us. And then he feels Australia is really poised to start creating these factories that make these vectors and reaping the intellectual property that come from them, and producing an industry for Australia, because we are well poised. And this is my last slide, and I really get a kick out of this, because as I said right at the beginning, mea culpa, I wrote a stem cell book, I wrote a genome book, haven't really delivered. Alan Townsend, he's the reason I wrote the stem cell book. So Alan Townsend has been involved in many medical revolutions. He's the pioneer of IVF, and then a pioneer of human embryonic stem cells. He got headhunted to go and lead California's $3 billion Institute of Regenerative Medicine. He spent about 5 years there. And then he came back. He's close to 70. What does he do? Instead of retiring, he gets involved in the gene therapy revolution. His mate, Martin Perra, once said to me, Alan Townsend has a sixth sense of where the next big revolution is coming from. And I'm reminded of what Martin Perra said to me, because suddenly Alan is right in the middle of the CAR T-cell revolution. So he came back from California and immediately got involved in this. I thought, oh yeah, that's interesting. And now I'm taking another look. And this is the Convergence Science Network. Luan would like me to talk about convergence. Well, this is a great example of convergence. Everybody else is making these CAR T cells by taking them from the patient, taking their own T cells, sending them off to America or wherever, where the new gene is put in, and then putting them back into the patient. Well, Alan's got a different idea, which he started in this company called CarTherix, where he's the CEO. So his idea is to make an off-the-shelf product that anybody can use, that should be cheaper. You don't have to send your cells off to America or anywhere. And the idea is to use stem cells. They're called induced pluripotent stem cells. They are indeed a product of the stem cell revolution. What it means is you can take any old cell, a skin cell, treat it with a few factors and turn it into the equivalent of an embryonic cell that can do anything. But rather than taking your own skin cells and doing it, his idea is to find some very unique donors who have cells that will match about 50% of the population. They're like type O's, except super type O's. Their cells will match 50% of the population. So he's going to start with those sort of cells from sort of near universal donors, and they're the cells that he's going to turn into guided missiles. And he's targeting the big game, which is the solid tumours, which so far are very hard to treat, things like ovarian cancer. And he will be using CRISPR, and he wouldn't tell me too much because it's all under, IP rules. But this is a wonderful case of convergence, and wow, Alan Townsend is back participating in his third medical revolution. And I said, well, Alan, what does it feel like this time? Because, I was interviewing him in the middle of the stem cell revolution, and that, this seems like it's already a much more present revolution than the previous revolution. Well, what does it feel like this time, Alan? And he said, it feels good. So that's Alan. He's got the laconic manner of a farmer, which in fact he is. That's how he started off, doing stuff with sheep. Anyway, that's it. Thank you very much.